And so we've developed technologies over the years to work with looking at collective behavior, so tracking large numbers of organisms, both in the laboratory and more recently using drones and deep learning technology, we're able to do so in the field, and I'll mention that in a moment. And of course, connecting theory and experiment is important, but we also need to develop new tools for data analysis, things like dimensionality reduction. How do we automatically find clusters in these complex data sets? And so that's another key component. And so those basically underlie the core of what we're doing within my department. And so the common theme among these very different systems that we study, I think it's over 60 species we study, the common theme is collective information processing. So when one looks at animal collectives, such as these schooling fish and flocking birds, unlike in the physical world, there's a real element here of information, of reduction of uncertainty in the environment. And one can look at different systems, such as these are blind army ants. It's literally the blind leading the blind, and yet they can construct scaffolding, they can construct bridges out of their own bodies to collectively solve problems. Here, the organisms are highly related to each other, but for many other systems, that's not the case. And increasingly, we're connecting our work to what's been done in neuroscience, such as the classic random dot paradigm for monkeys, where you have only a small proportion of dots moving, and you want to determine how the monkey, whether the monkey can actually detect that. So we can modify these types of designs to actually allow us to ask similar types of questions. So here there's 100% coherence, but it's the only video I had. And we can adapt this to ask about collective information processing within animal groups. So really trying to understand the fundamentals of cognition within these types of groups. So one of the great challenges is one of the reasons why collective behavior has kind of lagged behind other aspects of animal behavior is, as you can imagine, it's very difficult to get quantitative data from all of these animals simultaneously. So we also developed deep learning methodology that allow us to track animals and maintain the identities of hundreds of animals simultaneously without requiring marking of the individuals. We can also do things like raycasting to reconstruct the visual fields, so we can reconstruct the sensory information that the animals are using. And these technologies work with a very wide range of animals, from rodents to fruit flies. And a human couldn't see any difference between these animals, yet the software can. And we also develop real-time visualization tools as well. And so this software is much, much faster, 35 times faster, and requires much less memory than previous technologies to do this type of thing. And so returning to the central scientific question, it's really this relationship between individual and collective computation. We often tend to think of the brain as being the seat of intelligence, and of course that's partly true, but the interactions among individuals also give rise to forms of collective intelligence. And it's that ability to actually determine the sensory input. So the animals we use are often highly visual. They're not using the lateral line, the pressure sensing system, to detect each other. They're using vision. And so we can use these types of methodologies to infer the interaction network and to understand something about computation in the collective. So to give you one example of how things can differ between collectives and individuals in isolation, what happens if the world becomes more dangerous? Your prior is that the world is now more dangerous. Well, what we see is that these cascades, these escape behaviors, and we can trigger any individual within the group as we want to exhibit what's called a C-start. It's a reflex escape behavior, and observe how it spreads across the system. Well, the groups change their signal-to-noise ratio. They become more sensitive. They become more capable of propagating information when they perceive the world as more risky. Now, if you're thinking about an individual brain, how it responds when the world is more risky, well, animals tend to accumulate evidence up to a decision-making threshold and then exhibit a behavior. And so you may think, well, if the world becomes more dangerous, what the animal should do is to reduce this threshold so you make faster decisions, because making a false alarm is better than getting eaten by a predator. So you make fast but perhaps less accurate decisions. But no one has tested that within the concept of collective behavior. This is true of individual behavior. But if you think about the fact that these animals are embedded within a collective, there's another possibility. It's not mutually exclusive, but they could actually change the network of communication, and that could change the information processing capacity that we see at the collective level. And so when we ask this question, what we actually find is that the individuals, if anything, they increase their threshold. So they become less likely to make false alarms. There are more of them to detect potential risk. But what they do is they change the structure of the network of communication. So if risk is correctly perceived, it will spread very rapidly across the group. So they're doing something, if anything, the opposite of what you'd expect an individual to do. But because they're in a collective, it's the network of communication. And using the visual field reconstruction and machine learning, we were able to infer which visual features they actually pay attention to. And so these networks are not just schematic networks. The edges here, the weights represent how strongly I will change behavior if you change behavior, and the colors represent how reciprocal this interaction is. And we can now predict very precisely how far information will spread and who will be infected by behavior at a specific moment in time by understanding these processes. And what we've discovered is that computation is an emergent property of the network, the structure of the network, in a very wide range of different important ecological contexts. For example, regulating social contagion, modulating behavior in response to risk. We've also been able to show that it's a complex contagion process, much like in humans. Also sensing long-range environmental gradients. The individuals can't sense the gradients, yet via this network of communication, the group is capable of sensing the gradients. And this has then informed new algorithms for computational search and also in robotics. And finally, these are unrelated organisms. And if it's group level properties that are really important, how does natural selection then operate? Because the individuals, the selfish, genetically selfish individuals are the unit of selection here. And yet we've been able to show that these types of collective computations readily evolve among genetically selfish entities. And so this is a bit more reminiscent of how neuroscientists tend to think about computation. You don't just focus on the individual neuron. Yes, it is capable of quite sophisticated computation, but the network of communication among these neurons is also important. Changing the weighting and changing the structure of this network is important. And yet people haven't looked at that except until our studies in the context of collective behavior. So here we're studying animal collectives, and it's already beginning to make us think a little bit about the brain, and I'll come back to that again later. Okay, so I want to talk about some old work first, because it's going to set the scene for some unpublished work that I want to talk about in terms of the brain. And I'll start with thinking about leadership and collective decision-making in animal groups. And I'm going to show pictures of fish because I happen to have them from Matt McHenry, but you can think very broadly. Later on, I'll show you data from primates. You can think of bird flocks. I'll also show a little bit of bird flocking. So these models are very generic. And so what we assume is that this individual, how does the group make a unanimous decision? Say this individual here with the red arrow, that red vector represents its personal information. It could be that there's food over there that it knows about that the others don't, or maybe it's performed a migration before and the others haven't, so it knows where to go, the others do not. Does it need to signal to the others, I know what's going on, follow me? Well, there's no evidence of that in most of the systems that we study. We have studied the evolution of signaling too, but not going to be in today's talk. Do they need to recognize who has information and who does not? Again, we have no evidence of that. And so we'll start with a very simple model to ask how can information be conveyed in the absence of these complex mechanisms. So what we do know is that individuals are responding to others. They have social vectors pointing towards other individuals. And so the hypothesis here is that they then reconcile these social vectors with their internal goal-oriented behavior. And so if you imagine there's a group of 100 here, they start at random positions, random orientations. You don't know if anyone is informed or uninformed, but I'm going to color the informed individuals white so we can see. It's only so humans can see, right? So there's no way of knowing. And what we can find, the green cross just shows the centroid of the group. If these white individuals have a common preferred direction of travel, even with a relatively small proportion of them, they can guide the group highly effectively without signaling and without individual recognition. And then we can test these types of ideas with experiments. So these are real schooling fish. The red ones are informed, the blue are uninformed. Fish have amazing memories, by the way. You need to train them about 6 or 7 times and they remember for months. And so here you can see the information being conveyed across the group. And so we can relate, as we predicted from our theory, as you increase the number of informed individuals, you increase the accuracy of information transfer steeply, and then it plateaus. But what I find even more interesting is that, like us, like humans, different individuals have different experience of their world. They live in what are called fission-fusion populations. They're constantly merging and going off with others and coming back together. So you don't know what the informational status of others is. You only know your own informational status. And there can often be cases where there's a disagreement. Some individuals want to go one way, some want to go in another direction. How can the group come to a consensus? Because if you split, you lose the benefits of group living. And in the species we study, it's quite rare for them to actually split in this way. So how would we do that? Well, I would maybe ask you a question, like which cafe is the best cafe to go to this evening? And some of you would have one preference, some another, and I would do a calculation and pass back the answer. But these animals, to our knowledge, can't count. In fact, we've got good evidence that they can't count. They're just using these simple local rules. So how can they make a consensus? Well, what turns out to be important are the number of individuals that have different preferences, the strength of those preferences, but also the geometry of the problem. So I'm going to discuss the condition that almost all animals face, which is making decisions on the move. And so what I'm going to do here is keep one informed subset to always want to go 0 degrees, so along the x-axis. And then another subset is going to disagree with them. If the disagreement is 0, then of course they're not disagreeing, it's the leadership model. But they might disagree by a little small amount, by say 10, 20, 30 degrees, all the way up to 180 degrees on the far right, where they actually want to go in opposite directions. So these dotted lines represent the 2 different directions, the conflicts. And for each angular difference, I run thousands of simulations, and I'll show the probability of where the group goes. And the white line here is the average. If the individual could somehow calculate the average direction, it would fall along that white line. And so what we find is that below a critical difference of opinion, below a critical angle between the options, the group will move in the average direction, right? Even though they don't know what the average is, the group will move along that average direction. Now imagine they're really going to a red target and a green target in space. As you are far away, the angle is relatively small. As you get closer and closer, the angle will increase. And once you reach that critical angle, now it switches. There's this sudden bifurcation, and it switches now to a consensus where either half of the time the whole group goes this way, or half of the time the whole group goes this way, because it's symmetric. There's an equal number with equal strengths of preferences. And here I'll just show you a simulation. So you can see the sort of local noisy nature of the model, and it's going to be random which they choose because there's an equal number of white and red individuals. In this case, they choose the red. But if we ran that again and again and again, it would, on average, be 50-50. But what if I put just one extra individual into the group? Remember, they start at random positions, random orientations. You don't know if anyone agrees with you or disagrees with you. And it's sort of local, noisy interactions. Well, now what we find is below that critical difference of opinion is exactly the same as before. The group will adopt the average direction. But now once we reach that bifurcation point, that sudden transition, now almost always, 98% of the time, the group will spontaneously select the majority preferred direction, even though no one can count. And so here now there's an extra individual in the white category, and so they will almost always move in that preferred direction. So even though the individuals are using local noisy rules, the accuracy is extremely high. Now, when I wrote the paper, I was sort of quite rushed by the editors, and I was sort of writing, isn't it amazing that they can make this incredibly sensitive decisions despite all of those dumb, uninformed individuals, those green individuals there. It was only later that I realized that it's actually because of the uninformed individuals that they can make these decisions. Without uninformed individuals, groups cannot come to a consensus. They cannot even decide between two options. And so this kind of got me interested generally in the role of sort of uninformedness. We always think of being uninformed as being a bad thing, but here we show that a small proportion of uninformed individuals promote democratic decision-making within these groups. And there'd been some very interesting work by Bill Bialek at Princeton, where I was at the time, showing real connections between spin system models, like the Ising model, and neurobiological data in the cortex. And they had a beautiful paper in Nature with Ella Schneidman as the first author that showed that this really isn't just a coincidence, that there's really good reasons to make these connections. And so I wanted to ask whether this principle could be something that also occurs within the brain or within other types of systems. So here we looked at spin systems and showed the same types of results, that uninformed individuals promoted magnetization, promoted consensus within the system, and could reinforce the majority preference. And so, I used to think that this connection between how the brain works and how neural dynamics work and how animal collectors work is a metaphor. There's surely too many differences between these systems to actually claim otherwise. But now I'm not so sure. Now I think we genuinely can go beyond the metaphor. I think that natural selection has found algorithmically the same principles at different scales of biological organization. And that's what I want to discuss today. So this is now unpublished work. It's in revision at the moment. So I want to ask the question, how do neural collectives or individual brains make decisions? Because I realized that I've been thinking about these vectorial integration problems, and that's really what the animals are doing as well. Whether you're an invertebrate or a simple or an advanced vertebrate, you have to make sense of this vectorial world. And not only that, there's just been some remarkable work in the neuroscience community from people like Vivek Jarayaman, looking at the Drosophila, the ellipsoid body, and the navigational system in Drosophila. An amazing work by the Moses and others looking at rodents, and Nachum Malinovsky's work on bats is also absolutely remarkable. All of which shows that that we now know the genuine vectorial basis of decision making. So this is not speculative anymore. You can really find neural groups that encode information vectorially. So I want to think about this vectorial representation in the brain, both of insects and vertebrates. But to study this is quite challenging, doing conventional experimentation. So to do so, to really play around with space and time in new ways, we created an immersive holographic virtual reality for freely moving animals. We obviously can't put headsets on juvenile zebrafish that are less than 1 centimeter long. But what we can do is exploit an old illusion called the anamorphic illusion. And I can tell you right now that this tape doesn't have a volume above the table. And even if I tell you that, your brain still believes it to have that volume until the illusion is broken, and you'll see it never did. So this illusion is so strong for complex animals like us, we speculated that it might also be very strong for simple animals like insects and fish. Again, similarly, this shoe is not actually three-dimensional, but our brain believes it to be. And there's this nice demo on YouTube here, where if you track the camera, which is like tracking the eyes of the animal, you can show how we can then represent the world to be really immersive. You can move in and interact fully with these objects. And so that's what we did. We created this virtual reality system where we could have like a real zebrafish and a virtual zebrafish. And it's, we could control for distortion. We track the animal at 100 hertz. We project at 100 hertz. So we have a very high accuracy closed loop feedback. And then here, there is no pillar here. The world looks a bit weird to us, but it's always projected like that shoe or like that tape to be in the volume with the fish. And the fish will avoid it, even though it's not actually there. For the purpose of social behavior, we also create photorealistic virtual larval zebrafish. So we can show, for example, here, that even if this larval zebrafish is moving just in a simple circle, the real fish behind it, following it, believes it to be in the volume with it. We've really fooled the animal into thinking that there's a real animal in there with it. And this is much more effective than robotics, for example. But there's one problem with these types of systems, which is that I can't put more than one zebrafish into any of these tanks, because even a slight difference in position means that the world will look distorted to the second one. We always have to project the world from the perspective of one individual to have this illusion work. But what we can do, and what we do is we can have multiple systems. We have 6 of these systems. We network them together so the individual in the nearmost arena can interact live with a hologram of the individual in the second or the third, and vice versa. And so here you're now seeing 2 individuals that are interacting not in the same physical world, but in the same holographic world. And we call this the matrix for obvious reasons. And of course, we've taken great care to establish that the way that they interact, the mechanism of the interaction, is absolutely identical in the real physical world. So here's the matrix. This is at the top, the turning forces that individuals exhibit when others are in certain positions. Below are the speeding forces. And if you compare that to then taking the 2 fish and putting them in the same physical world, we see extremely similar data. The reason the physical world data is not quite so clean is that the 3D tracking system sometimes makes mistakes when they get too close in the real physical world, which is not a problem in the virtual world. But the structure is identical. So everything we've done really shows that we're completely fooling these individuals. And so, as I said, we have up to 6 of these systems. Here are just 4 individuals interacting within the matrix. We can put in arbitrary complexity in this environment. We could put predators, could put different environmental conditions. It's really up to the imagination. We also, we can play around with space and time in ways that you can't in the real physical world. But because I got really interested in how the brain is making decisions, we also built a virtual reality system for Drosophila. I never thought I'd be working on Drosophila. And also for terrestrial animals like swarm-forming locusts that we also study. This is a massive, huge motion compensating sphere. So there are many advantages here. We've got this very high degree of control over the inputs, the sensory inputs. So we're focusing on vision. We can quantify individual differences. But really what we can do is we can really explore parameter space in a systematic way. And that allows us to look at the algorithms that the brain is implementing. And that would not be possible in conventional experimentation. And as I said before, we can also play around with space and time in ways that is not possible in the real physical world. I won't go into it today, but this has shown us very good evidence that the brain is not responding to space in a Euclidean way. It's a non-Euclidean way that they respond to space. So coming back to the scientific question of vectorial representation in brains, really it's like what I showed you before, but here the vectors are not different individuals within the group having preferences, but that there are different neural groups within the brain that have different preferences, and they're competing with each other to make a consensus decision about where the animal should go. But it's the same principle. And I won't go through the details of the model here, but we use a very simple model where there's reinforcement or excitation among neural groups that have a similar preference, and then inhibition between groups that have a different preference, or actually you can just have global inhibition and the results are near identical. And this type of model, for those sort of familiar with this, is called a neural ring attractor network. And it's a quite commonly used model in neurobiology to understand these types of processes. Now, we use a very simple implementation of that because we want it to be analytically tractable. And I think sometimes, neurobiologists, because of the amazing work that's been done at sort of mapping these types of networks, there's a tendency to think that these details might be very important. And indeed, I think they are very important for certain things like predicting optogenetic experimentation, for example. But there can also be, if one builds a very complex model, like this one from Ben de Behaviour and a colleague, where they try to put in a lot of this detail that you see within the structural connectivity, what emerges at the end is actually a model that is no different in its dynamics to a very simple ring attractor. That not many of these details do not matter. I'm not saying it's not important to look at these details. I'm definitely not saying that. But I think there's a real validity to starting with a very simple model. So in this paper, for example, they found that their model behaved like a ring attractor under a very wide range of parameters. And that this was robust to physiological details, such as variation in synaptic weights. And they wrote here that the ring attractor computation is a robust output of the circuit apparently arising from its high-level network properties, topological configuration, local excitation, and long-range inhibition, rather than the fine-scale biological detail. And also, this ties in with another branch of mathematics where these ring attractor models have been studied from a mathematical perspective, and that again has also shown that the solutions do not require specific simulation of neuronal dynamics. There are families or classes of models. Okay, and this again is how physicists often think about these types of systems. And so while capturing known generic features, these topological features and excitation, local excitation and long-range inhibition, our model is deliberately minimal. We're trying to find the simplest model that can explain and predict observed phenomena. And we also, I'm not trying to make a model of the Drosophila brain. I'm trying to make a model that captures essential components of all brains. And also, it's analytically tractable, so we can really understand why we see what we see. And of course, we can start simple and add complexity in. There's nothing stopping us doing that. But let's start with a very simple model, the simplest possible model of neuronal dynamics and what it can predict. Now, when I started working on this, I thought, there's hundreds of papers where psychologists, biologists, neurobiologists have reported the outcome of decisions, what an animal does, whether it chooses option A or option B. But I could not find a single paper where they've reported the trajectory of the animal as it makes that decision. It's almost as if people think that the animal makes a decision and then the movement is just the output to facilitate that decision. Rather than what's very important here is the recursive feedback. An animal moves, it changes the sensory input, which changes the movement, which changes the sensory input. And biologists often just put the animal in between, directly in between two options. And I'll show you why that is such a bad idea in a moment. But first, let's look at what the model predicts. So if we think about an animal moving towards two targets initially, two identical targets initially, theta here shows the angle between the targets. Remember, when the animal's far away, the angle will be relatively small. As it approaches the targets, the angle will increase. And this is the analytic model showing that there exists a bifurcation, which is that solid black line where the brain should switch from averaging those inputs to deciding between those two options. Very reminiscent of what I showed before with the model of animal groups. The gray region shows a hysteresis region. So, and this depends on neural noise. In the hysteresis region, once the brain has made a decision, then it doesn't flip back to indecision. So it could make a decision for any of those angles within that gray regime. And so if we look at the simulation in this case, actually, the more neural noise you add, actually, the more deterministic you can define this critical point. But here, the prediction is that the animal is moving towards two static targets. So in our experiments, these are pillars for the flies and the locusts, and they are naturally drawn to those. It's a reflex fixation. We predict that the animal will move until this critical point, and that's this quasi-phase transition, and then we'll randomly choose between the two options. And so why is this important? Well, it's important because it really reinforces this idea that there's a recursive mechanism at work that's essential to decision-making. But also what we know about critical points, doesn't matter what the system is, it could be an ecological system, a physical system, there's a universal property at the critical point, which is that a feature called susceptibility goes up. Susceptibility is how sensitive the system is to any differences between the options. And it's not something you put into the model, it's an emergent property that is inevitable for any model that exhibits a phase transition. And so at this critical point, we show in our model that the animal should become spontaneously extremely good at discriminating between those options. And I just want to stress that we didn't add this in. This is an emergent property. What a wonderful thing for a decision-making circuit to have, which is why we predict that they've evolved this capability to go through bifurcations. Because now if there's just a slight difference in neural activity, even though these neurons are noisy, even though there can be relatively few neurons in a network, the system at that critical point becomes extremely accurate, right? So the systems can be noisy as hell, but at the critical point, it will always be extremely accurate. And also, depending, the animal can come in from any different angle and will reach this critical point and will benefit then in its decision making. So that's the theory, that's the prediction, that there should be this bifurcation dynamic. I'm not a neurobiologist, so I would have gone into the brain to look for this. But we realized that there's a prediction in the trajectory space, in the space-time of animal movement. But as I mentioned before, I couldn't find any data of this. So we did our experiments with fruit flies, and what I'm showing you behind is a density map of hundreds of trajectories, and also here with locusts moving towards 2 static targets. And as you can see, we found strong evidence that indeed the brain undergoes this bifurcation. It moves along the average, reaches a critical point, and then the brain decides. But what was even cooler and kind of unexpected to me is what happens with 3 options. I figured that the rightmost option would cancel the leftmost option and they would just go down the middle. And that indeed is what all existing models, vectorial models, including my flocking model, by the way, does. But the theory here predicted something different. It predicted that if we're correct, the brain spontaneously breaks this down into a series then of binary decisions. And at each critical point, the brain will be incredibly sensitive to any differences between the remaining options. So it turns this complex world into a series of binary decisions. And also, I'll show in a moment, works with any number of decisions. It's very hard to do these experiments, it took many, many months. But we did these experiments, and indeed the brain breaks the world down into sequential binary decisions, as predicted. We can also show this works in 3-dimensional space. It works with any number of options. So it spontaneously solves all of these problems. But if this is a general principle, then we should also find it in the vertebrate brain. Even though those 2 insects are not highly related, the vertebrate brain is, of course, very, very different. So we turned to larval zebrafish to understand how they make decisions. And again, this involved our virtual reality environments. You could never do this precision-type experiment in the real world. And here we have a slightly different ecological scenario, a scenario of schooling behavior, where the individual, the real individual, will tend to school with virtual individuals. Here, just for convenience, shown in red. And if we change the lateral distance between those virtual individuals, which would be something really hard to persuade real fish to do, but across a biologically meaningful range, the model predicts again that the brain should exhibit a bifurcation. And this is what the data show. Indeed, the brain is exhibiting a bifurcation. If we have 3 moving options, again, the model predicts, by the way, this is not just the same model, it's the same model with the same parameters. Right? That's how robust the model is to these types of features. We predict this double bifurcation here, and it looks a bit different because of the moving frame of reference. And then looking at the experimental data, that's indeed what we find. And we would never have known what experiment to conduct or been able to interpret this type of data had it not been for the guiding of the theory. We've also looked, for example, at asymmetric geometries. Most models of vector integration would say you've got double the probability of following the 2 versus the 1. Our model predicts it should be roughly the same. In one case, you're below the bifurcation, and the other, you're above it. And we could predict very precisely the peaks of the distribution and also the symmetry of the options that individuals exhibit. We're also able to show that things like the optomotor response that were thought to be involved in schooling are not. It's a completely different system. And so now working with Armin Ball, who's recently moved to Konstanz from Harvard University, we're now really being able to get in and understand the neural dynamics. So Armin is an expert at really dissecting these circuits for evidence accumulation in the zebrafish. So I find that extremely exciting that I'm now able to sort of work with him and get into the brain to understand how this is actually modulated. But another really exciting thing is, in studying the brain in this way, I suddenly realized that all flocking models are wrong. If you actually go back to all classic flocking models, people haven't ever looked at 3 options before. We know real animal groups can choose between 3 options, believe it or not, what a surprise. But all classic flocking models, including my own, are not capable of doing that. It's lacking a fundamental feedback. And so now studying the neural dynamics has allowed us to go back and make predictions about how that feedback is implemented in animal groups and how this should be recovered. We can then also use new technologies. So this is work with my PhD student, Ari Strandberg-Peshkin, who's now a group leader in Konstanz, Damien Farine and Meg Crowfoot, who's co-director of that institute, where we put GPS collars and accelerometers on almost all adult baboons in a wild troop in Kenya. This is a huge investment of time and money to do this type of work. It's very, very difficult to do. And obviously, for ethical reasons, we did not put collars on juveniles, and all collars were removed at the end of the experiment. This allowed us every second in their lives to know where these baboons were, and even to infer more than that from the accelerometry data. And what we realized is that in this rich data set, there were tens of thousands of events where some individuals want to go one way, some individuals want to go another way, and yet the group will almost always come to a consensus about where to go. So we were also able to ask about the brain of the baboons. These are the predictions, of course, about the bifurcation dynamics. And we could show that indeed, there is evidence that the brain is also behaving in the same way for the same reason. So these very robust geometric principles really span scales of biological organization, from very simple animals to highly complex animals, and even to animal groups. Even though the feedback is mediated in a different way in the animal group, it is there for the same reason computationally. So there's a sort of fractal nature of this, this algorithm across scales of organization. We've also worked closely with Martin Wikelski, who's an expert in bird migration. And these are real data. We can even get the flapping behavior of these storks because we have these accelerometers can tell when the birds are beating their wings or when they're gliding. And here we've also been able to show that the behavior conforms to our models across, in this case, a massive scale. We catch the animals near Konstanz, and they migrate across sub-Saharan Africa. And so we can now look at this detailed data of the individual flapping, and we can reconstruct the thermals, and also look at the decisions that they make to choose where to go between thermals and how they utilize social information. So it's an incredibly exciting time to do quantitative behavior, both in the lab using things like VR, but also in the field using these new types of technologies that Martin and colleagues have developed. But there are problems. I mean, that's extremely expensive to buy these collars. You have to get a whole team of vets and scientists out there. Here's a recent field trip just before COVID I took with us, Bill Hansen, for anyone that knows, the ex-vice president of Max Planck. We were studying buffalo, and this again involved a huge team. And we also had visitors like elephants that you need to keep your eye out for. So great fun, very important, but really expensive and impractical. There was also a helicopter and all that kind of stuff. I can't afford that sort of thing myself. So we've decided to go cheap and use drones and try to really push massive multi-scale imaging using cheap commercial off-the-shelf technology. So we started this some years ago using more professional drones like the SenseFly drone, but now we can just use off-the-shelf DJI drones and reconstruct to about 3 centimeter spatial resolution the structure of the habitat through which animals behave. If we look at a 2D projection, this is a 3D habitat, but a 2D projection of it. As we zoom in here, you at first think, well, yeah, there's some animals moving around there, and it looks very much like you get from GPS data. But as we zoom in further, we actually have a group of drones that collect these data. And you'll begin to see that this is sped up. You see the little tails moving furiously. So we've developed this markerless tracking technology, such as also being developed here in Tübingen, that allows us to really get extremely detailed information. And we would never get permission to catch and tag these animals anyway, because they're an endangered species of zebra. There are only around 2,000 left. So the species that are most vulnerable to humans are the ones that we're least likely to be able to get quantitative data for using conventional methodology. Yet here we have a non-invasive methodology where we can even identify individuals and get much, much more behavioral data than you would get from GPS. So in the last couple of slides, I just want to talk about building collective behavior in Konstanz. One of the other reasons I was excited to come here is to talk to the Excellence Cluster, UFOC, because we also won one of these clusters, and it's been really interesting and exciting to talk about the similarities we've had actually in our experiences. And it's been wonderful for us at a small university like Konstanz to be able to bring in computer science, physics, biology under this common area, whether you're interested in cells or whether you're interested in physical particles. So we have this true interdisciplinary team Konstanz. And also in addition, thanks to Hector and the German government, we were able to also get a building for the study of collective behavior. This came before the cluster, and it's just opened 2 months ago. And it's a really, really nice place to be. So please do come and visit. We are now open for visitors, and this is the view from our building. So we would love to see you. And again, directly inspired by the Max Planck Institute of Cybernetics that I visited when I came here, we've built a huge imaging hall for 3D tracking, sub-millimeter precision, 300 hertz tracking of animals. Except for Tübingen, this is the biggest place in Europe to do this type of study. And so here you can see we can track not just the positions and motion of the birds, but precisely where they're looking and reconstruct the world from their perspective. And we also collaborate with psychologists in this environment, in our case, looking at eating behavior, how people influence how each other make eating decisions, but sort of bringing this technology that I say directly inspired by my visit here to Tübingen many years ago to Konstanz. And we also have a new Max Planck Institute, so which we're just starting. We don't yet have a building, but we do have 3 directors. And we got 60 million euros to build a building on the campus, and that's just currently in the architectural stage. Thanks very much for listening. Oh yeah, we've got open positions as well. Thanks a lot for listening.